research. Then he switched to cancer research in his PhD studies and studying the metastatic dissemination, identifying clinical useful predictive markers, and developing novel therapeutic tools for breast and gastric cancer at Weill Cornell College of Medicine. And he was recruited to BC Cancer Research Institute in 2021. And now he's studying identifying and understand the mechanism that drives the progression and dissemination of B-cell lymphomas, as well as developing a minimum invasive diagnostic tools and novel prophylactic and curative treatments. And so I'll give the floor to Leandro. Please go ahead.
Thank you very much. I'm going to share my screen. Are the slides showing okay?
Yes, it's good.
Okay, perfect. Okay, well, thank you very much, Dr. Gao, for the invitation and everyone who joined this call. As Dr. Gao was saying, I'm a scientist with BC Cancer. My group started almost 3 years ago, and we have a keen interest in B-cell lymphomas. So other than talking about the work that we're doing in our lab, I thought it would be a good opportunity maybe to contextualize a bit more and discuss more in general what has been happening and what will be happening in the field of B-cell lymphoma modeling, and how that can really help drive the clinical side of things as well. I'd like to start by acknowledging that we live and work in the ancestral and ceded territories of the Coast Salish people, including the Musqueam, Squamish, and Tsleil-Waututh nations. So the 3 big topics that I'm going to be talking about today is first, just to bring everyone on board, I'm going to be discussing some of the challenges that remain in the clinical management of B-cell lymphomas, and that's coming from a PhD. The second one is discussing about what are the different strengths of limitation of classical and more modern approaches to generating preclinical models in lymphoma. And then I'm going to be focusing a bit more on illustrating how genetic engineered mouse models, or transgenic mice, as a simpler term, can really help us push forward our understanding of this disease and its targetable biology. Because that's one of the things my work has, my lab has a keen interest in. So just a very brief introduction, a few numbers. Lymphoma is actually a very broad term that basically, or an umbrella term to serve to group a lot of different malignancies originated from lymphocytes. Annually in Canada, there are approximately 13,000 new cases and about 3,000 deaths, unfortunately. Going from higher to more refined classifications, the broadest 2 categories that we can find are Hodgkin and non-Hodgkin, with the second being more frequent. Median age of diagnosis is about 40 years old for Hodgkin, but there are also childhood and adolescence forms of the disease. And for non-Hodgkin lymphoma, the median age of diagnosis is a bit later at around 70 years of age. Survival rates are fairly high survival rates, but that's really masking a lot of heterogeneity. Because as I was saying, when we talk about lymphoma, we're talking about many different diseases, and that includes many different histologies, different localization for the tumors, from the lymph nodes to virtually any organ that we can find. There are different behaviors, so we can find, let me point my pointer. So we have forms of the disease that are indolent, that are gonna grow and advance for many, many years. And then we have aggressive or very aggressive variants of the disease. And of course, there's, within all of those categories, there's a lot of biological heterogeneity that we and others are trying to capture by using molecular classifiers. So each lymphoma type, of course, has their dedicated approaches and regimes to treating, but just to get a general picture, because this could be a whole talk or many talks in itself, approaches are used commonly in lymphoma include the watchful waiting, especially for those indolent forms of lymphoma. different regimes and combinations of chemotherapeutic agents, radiation, and something that where the lymphoma field has been at the forefront is implementation of targeted therapies, and that includes anywhere from monoclonal antibodies such as rituximab and an anti-B-cell antibody, to immunomodulatory agents, checkpoint inhibitors, and more recently T-cell based immunotherapies. The approach that is not really captured here is stem cell transplantations that usually reserved for more cases of relapse or refraction to therapy, and that kind of take the form of either an autologous or an allogeneic stem cell transplant. So we have all these arsenal or collections of therapeutic agents at our disposal, but that doesn't mean that we're far from having solved all of our problems. So what are those big issues? And we can really identify challenges all the way from the early detection and diagnosis of the disease and all the way to treatment. So just to highlight some points, when we talk about the detection and diagnosis, we don't really have a lot of tools or very limited tools in terms of achieving an early stage detection of the disease. And even at the time of diagnosis, it's still challenging for some entities to define exactly which category will be in the tumor and how to assign a new patient to all of these categories. And that's going to influence, of course, how we manage the disease and how we treat it. In terms of disease stratification, the tools that we have are useful, but not great, for example, in telling us which patients may advance or transform from an indolent to an aggressive form of the disease, or which patients are most likely to experience relapses. Moving to treatment, some of the challenges that are probably common to other types of cancer are second and third line of therapy. Of course, as the patient progresses, we have less and less effective tools, including even when we consider immunotherapies. We don't really have a scheme of prophylactic interventions for patients. I think we have to consider aspects such even for the existing therapies, if we have elder patients, they won't be suitable for all of the approaches that we have at our disposal. Some tumor types or some lymphoma types are still considered incurable entities. And then aspect that I'm definitely not going to touch on today, but are important, have to do with the frequency of monitoring and regulatory and financial aspects of treatment. So many of these aspects probably we could start to find answer if we had a better understanding of the disease, and there's more than one way to go about it. So one that's been incredibly helpful and informative has had to do with direct profiling of tumor or clinical specimens, and the BC community, many in this call today, have had a major role in us understanding the biology and the clinical behavior of tumor. So even from more than 20 years ago, people around were characterizing expression of oncogenes in lymphomas, then extending those assays to studying the genomes of the tumors, epigenomes, transcriptomics, the immune context in which these tumors may arise and provide risk factors, all the way to more recent advances related to spatial features of the disease, and of course, a lot of valuable work on the clinical behavior of these tumors, including clinical trials, retrospective studies, etc. So all of these approaches provide very, very useful insights, but they really can't provide all of the answers that we need if we're to tackle these problems. So an alternative approach to addressing these areas of need is to turn to lymphoma modeling. So I'm going to go over the next half an hour or so into each of these categories, but in a snapshot, basically, when we talk about lymphoma models, we're including anywhere from a primary tumor cell from the patient to a cell line, to cell lines organized in 3D cultures, to systems where we graft cells, immune-deficient mice, and also using transgenic mice to mimic key aspects of the disease. So a phrase that I really like, and as someone working on modeling hits very close to home, was coined by a statistician many years ago, and it was referring to statistical models, but I think it applies to anyone doing modeling science, which is all models are wrong, but some are useful. So before we go deeper into what each type of model can offer for us, I'd like to highlight some areas where this definite modeling can be complementary to all the profiling that can be done in clinical specimens. So for the first part, dealing with risk factors. So models are very good or very useful in terms of deconvoluting the contribution of the genotype versus the variable of time or aging and the immune history of the patient. It can also directly help us study which genetic alterations might have an actual driver role from which those that might be a passenger effect. Something unique to modeling is also, so it's very useful to backtracking the earlier stages of disease and how they may evolve and advance into a tumor. So that's, of course, critically relevant if we're thinking of down the line developing early detection approaches. And finally, for therapeutics, they offer possibilities, for example, to test something as simple as testing multiple doses and combinations that would not be feasible to do in clinical practice. They allow us to test new candidate targets, even when we may not have a usable drug for them, at least as a proof of principle. And they could be particularly useful for example, for rare form of lymphomas where doing a clinical trial is almost close to impossible. So if we can only do a few clinical trials a year every few years, these models can help us prioritize the most promising approaches. So probably the simplest models are cell lines. And I think all of those probably on the call are more than familiar with those. So I'm not going to spend a lot of time on how this can be generated and used. These are just some examples of, and the list is way, way, way longer than this, of the arsenal of cell lines that we have corresponding to different lymphoma types. What I would like to spend a couple of minutes is highlight maybe or make it more explicit the pros and cons of each of these models. So cell lines definitely have very evident advantage that have to do with how easy it is to grow them in large scale. They're broadly available, they're broadly commercially available or from academic collaborations. They're fairly easy to manipulate. So if we want to mimic genetic events, that's going to be fairly straightforward. And from a regulatory requirement, they're pretty straightforward to deal with. Some of the obvious cons that these have as a model, of course, is that the cell line has lost after repeated passage in vitro a complete, like they become fully independent of their tissue after tumor microenvironment. They probably show different degrees of transcriptional, epigenetic, and phenotypic drift as corresponding to the original tumor. They tend to be fairly homogeneous. We know that actual tumors are super heterogeneous, whereas this is largely lost using cell line models. And other aspects are that not all type of tumors are going to have a proper model, and that they're not great at allowing us to study toxicity beyond the effects on the tumor cells themselves. That doesn't mean that they still don't hold value. So there's some approaches that are particularly well suited for using cell lines. One of those that is used a lot in the field these days is, for example, leveraging cell lines to do CRISPR screens. So the basis behind it is that we can target in parallel a lot or all of the genes in the transcriptome of the cell, and then add conditions where we treat with different drugs or expose us to different conditions or screen for different phenotypes at great scales. So some of the recent examples I wanted to highlight in how can we still find a use for cell line these days is, for example, to study why does a therapy work. So in this recent study, the authors found that the response to ibrutinib, BTK inhibitor, was actually acting through inducing an atypical autophagy program in cancer cells. Other groups have leveraged this approach to actually test why therapy does not work, and for example, use a CRISPR activation library in this case to assess what provides sensitivity or not, or resistance to radiation therapy. Some other groups have leveraged this to study off-target effects, and also, for example, to empower immunotherapies by exploring which genes in tumor cells are controlling the expression and function of MHC class 1 and 2 complexes that could then be used to revert the immune evasive effects in tumor cells. So cell lines are definitely still useful, but as we highlighted, have several limitations. So the question is, can we do better? So a possible approach is moving from cancer cell lines that are basically have been passaged many, many times in vitro to moving to more approaches that use primary tumor cells. So the first limitation that we find is why not just use primary tumor cells, where the response is for anyone that has ever tried to generate a cell line is that most primary tumor cells don't grow or don't survive well ex vivo, or sometimes they may survive but not divide and not persist to the extent that we need for experimentation. So not really a novel concept, but there have been many efforts over the years in the lymphoma field to develop lymphoid organoids. I do want to make a distinction that an organoid is not just a 3D culture. The concept of an organoid means that in addition to this 3D component, there's some sort of self-renewal nature to the structure, and that cells can also organize and reproduce key functional aspects of the organs or the tissue where they come from. So it's not just clumps of cells, but actually organization and function. So when we talk about organoids, there's a few, not all organoids are made the same, and there are basically three aspects that we can consider. So one is the composition of the organoid, whether it's using directly tumor cells and just artificial stroma, whether it's a combination of the both, or whether it's a complex mix of cells extracted directly from the tumor. In terms of the source, we can think of organoids made directly from tumor stemming from direct biopsies, or those that have gone through a xenograft approach, where we're going to discuss later in the presentation. And finally, the type of organoid, they're organized that are scaffold and are going to have different matrix components to mimic the context in which cells exist in the tissue, versus there are others that are basically free floating and there's no structure to them. Of course, each of these come from with their considerations. So when we talk about more defined mix or only using tumor cells, the composition of these organoids tends to be a more arbitrary, versus when we use a complex mix, it tends to be more like a black box approach. When we talk about sourcing primary cells can be definitely a challenge in itself. When we use xenograft, the offer of type of tumors may be more restricted. And in terms of engineering, of course, the scaffolding variants tend to have more higher costs associated with them, but also more complex to set. So again, going a bit now over the general pros and cons that we see with these models when using lymphoid organoids. Some of the advantages is that we can recapitulate to some extent the TME or the stromal components of the tumors. They can capture some of the tumor heterogeneity. They really lessen the need for animals, which would otherwise we needed to recapitulate some of these aspects. And they allow for a relatively fast turnaround from the time we obtain a biopsy until we can be experimented with these organs. Of course, some of the cons that we mentioned are getting the samples themselves. Then more lately, the switch from a fine needle aspirate to a resection biopsy has been challenging for securing enough number of fresh live cells. And then, of course, there are challenges related to mainly that these platforms are somewhat early, even in their early stages, I would say, even though people have been working on them for a while, the success has been quite limited. But we can look at least at two examples where these have actually been put to good use. So the first example that I'm going to raise is that this group of Patricia Perez-Galan working with mantle cell lymphoma. So mantle cell lymphoma typically grows in the lymph node. This is a type of lymphoma that has a very close knit interaction with their TME and a close dependency. So what this group achieved is basically to generate lymphoid organoids that recapitulate in marginal zone lymphoma by mixing tumor cells from the patients and then extracting immune cells from PBMCs from the same patients, so matched, mixing them in a non-scaffolded setting and adding key survival cytokines to supplement the growth of these cells. So this is what the authors show that their organoids look like with and without the media. what they could demonstrate is that the tumor cells themselves show active BCR and NF-kappa B signaling, which is typical in this type of lymphoma in primary tumors. They show that the transcriptome of their organoids resemble that of the primary donor, and they could show that the T cells in their cultures show features of exhaustion, which is also common to see in this disease. Perhaps more interestingly, what they showed is that their organoids could recapitulate the sensitive or resistance to ibrutinib, which is a BTK inhibitor that's used in refractory or relapsed mantle cell lymphoma. So here I'm just showing a panel from that paper where they show that if they prepared organoids from a biopsy obtained prior to treatment with ibrutinib in the patient, when the patient was a responder, they can see that their organoids also show that sensitivity, versus when they move to a biopsy obtained after the patient developed resistance, they can replicate this behavior. Now, what this means is that these are fairly decent systems to recapitulate what is happening in vivo, but they could take it one step further. And what they showed is that when they use nivolumab, which is an anti-PD-1 that's approved in other types of lymphoma, but not in MCL yet, and they combine it with ibrutinib, they could actually overcome the resistance that they were seeing in these patients. So these organoids can be used not only to recapitulate and follow the patient along, but they could also be used to predict what sort of new combination therapies could be beneficial for the patients. Another group that more recently or quite recently came up with a publication using lymphoid organoids was describing lymphoid organoids for follicular lymphomas. This is for the most part an indolent form of lymphoma that again recapitulates very closely the biology of germinal center activated B cells. In their approach, this group basically took the strategy of going for doing basically taking biopsies, preparing single cell suspension, mixing them all together, and not adding any scaffolding or external cytokines, but rather really letting them be. So this is basically what the lymphomas look under this microscope. And in addition to showing that the relative composition of tumor cells and other immune populations could be maintained over several weeks, they could also show that basically the organoids were clonally stable and that the mutations were somewhat stable over several weeks of culture, making them a good model because it's not drifting very quickly from the original tumor. Then again, these models were used to study immunotherapies. In this case, they were studying two bispecific antibodies, one with a domain that binds CD3 and CD19, and the other one that binds CD3 and CD20, both approved for the use in relapsed refractory follicular lymphoma. What they could find was that these antibodies in the organoid system did not behave the same, with the CD3, CD20 performing better. And they even were able to take it one step further and to correlate that these differences in responses among the tumor cells were not apparently related to the activations of CD8 cells, but rather to the activation of CD4 cells, which was an interesting and unexpected finding. So definitely there's a lot of value in this type of approaches. So as a quick partial summary of these ex vivo models, cell lines still definitely hold value, although with our attention gearing to more focus on immunotherapies for therapeutic approaches and research focus really nailing down on the tumor microenvironment, maybe the cell lines become a bit of a rest level model, but they still have a whole lot of value because of their ease of manipulation and ability to grow in large numbers. On the other hand, the modern approaches to constructing organoids with lymphoid tissues appear very promising, but probably still a bit early in their development, in that we haven't really seen that the community is really taking these approaches broadly. So the other type of models that we can think of now moving on to an in vivo setting is those that relies on grafting tissues from the patients or from cell lines into immunosuppressed animals. So the first category is that of CDX. By CDX, I'm referring to basically taking cell lines and implanting them in fully immunodeficient mice, and most commonly injecting them subcutaneously so that they develop orthotopic tumors. So they do show some advantages as in terms of using them as models. So the engraftment rate is very high and very predictable. They're fairly simple to establish, and the regulatory constraints are minimal for working with these models. On the downside, these tumors are still growing in immunodeficient animals, so we still don't have a proper tumor microenvironment or an immune component. We're still working with cell lines, which means that we can have a fairly big distance to primary tumors. And because they're grown from cell lines, they're still probably, for the most part, homogeneous. Again, that doesn't mean that they don't have any value. So at least some of you, I'm sure, are aware of the recent breakthrough in the field of, particularly in the field of diffuse large B-cell lymphoma, where recent investigations showed the advantage of using a new type of monoclonal antibody, polatuzumab vedotin, both in the refractory setting, but also in frontline for the treatment of frontline diffuse large B-cell lymphoma. So this drug is basically an anti-CD79, the antibody that's fused to an antimitotic drug. So basically, while many groups were investing a lot of efforts in conducting clinical trials with these drugs, at the same time, group already started looking using CDX models as possible mechanisms of resistance developed by tumors against this drug. So in this case, these two studies by the same group would really focus on, while the drug was still being revised by the FDA and it was under evaluation, they very quickly managed to develop resistant models to the treatment with polatuzumab and come up with possible combination strategies, in this case by using rituximab, another monoclonal antibody that's used in diffuse large B-cell lymphoma, to potentially overcome this resistance. So this type of models really allow us for a fast turnaround time and to mimic effects that happen in patients. A more sophisticated way or approach that still relies on grafting is that of PDX. So unlike the other approach where we're using cell lines and implanting them in immunodeficient mice, we're using directly tumor cells growing from the patients, from the biopsy, and going into animals that can then be subjected to serial passages. So they have more advantage because we're working now with primary tumor cells, so more closer to the organized system. They do show molecular heterogeneity. They show clonal dynamics that can be followed. And there are public repositories or commercial where one can source these type of models. The downside is that it's really technically challenging to establish these models. The engraftment rates for tumors are incredibly rare, it's very low, which means that it's hard even for some forms of lymphoma to have representation at all with these type of models, and really needs a very big set of infrastructure and highly trained personnel to maintain this sort of system. So there's not a lot of examples on using these models, but at least groups from the MD Anderson and from Harvard achieved to generate a fairly decent collection of these tumors. So in this case, I'm citing a paper from the MD Anderson group, where they were able to establish different lymphoma models for marginal zone lymphomas, mantle cell lymphomas, and diffuse large B-cell lymphomas. They could show, among other things, that the histology of the tumors of the PDX was similar or at least relevant to those in the primary human tissue. And they could show that even after repeated serial passage of these tumors, the tumors maintain their genomic stability. So again, the first step is to basically validate these models, but the next one is to wonder how can we use this for in a manner that's translatable. So in that paper, the authors provide this example where it was really remarkable because they had a patient suffering with mantle cell lymphoma. This was a very heavily pretreated patient, which, as you can see, had been given a lot of targeted therapies, radiation, also an autologous stem cell transplant. they were put on ibrutinib, developing or achieving 3 years of remission, but then eventually they relapsed. At that time of the relapse, the investigators took the biopsy from the relapse and generating a PDX model very quickly, which then allowed them to retrieve cells from that PDX and do large screens for different therapeutic agents that could maybe help this patient overcome the resistance. So by testing different agents, they came to the conclusion that bortezomib, to which the patient had already been exposed, could be beneficial for the patient. And so around 3 months after taking that initial biopsy, they could go back and basically select this drug for the patient, so it was already approved. And as you can see, indeed, now the patient was showing that very dramatic response to the therapy. So while this is probably very hard to achieve on a larger or broad scale, it serves as a proof of principle of how useful these models can be, not just to recapitulate what's happening in the patient, but also to inform next steps. So the question is, can we improve or build even better on this model? So one area of work that a lot of groups, even here at PC Cancer, are working on is on developing humanized graft recipients. What I mean by that is basically taking these approaches of doing either cell-derived xenografts or patient-derived xenografts, but now implanting, instead of a fully immunosuppressed mice to avoid rejection, to humanize the recipient, either by transferring hematopoietic stem cells or PBS that can basically reconstitute the immune system of the animal and replace it for a human-like version. So the pros of those models are probably very close to or in addition to all of the benefits that the PDX model has to offer, these also can recapitulate, for example, a full or functional T-cell compartment. There are still challenges with these models because they're still somewhat in early stages of development. Some of the difficulties that people or groups are finding is still dealing with low engraftment rates, with graft versus host disease, because of course the recipient and the donor need to be HLA matched. And also some of the challenges in at least the first iteration of these models was that the myeloid compartment was not fully developed or functional. That being said, over the last 2 to 3 years, we've had different examples of people using mostly cell-derived xenograft in this context to generate humanized mouse models of classical Hodgkin disease, Burkitt, DLBCL, and even using humanized models to infect with EBV virus and try to recapitulate how that contributes to the pathogenesis in B-cell lymphomas. So the last part of this talk, I'm going to focus more on the use and development of genetic engineering mouse model or transgenic mice. It's something that my group and of course others work very heavily on. So I think most of you are probably familiar with the concept of a transgenic mouse, but just to bring everyone on board, the idea is that we can take an egg, inject or use viral DNA or use CRISPR or different modern gene editing technologies, transfer to a surrogate dam, and then from there on generate transgenic offspring that can be bred between each other to generate the genotypes of interest. So these models offer a lot of flexibility in the type of things that we can model. So from even just a purely genetic side of things, we can model gene or full exon deletions. We can model gene gains. We can model replacement of genes, fusions, point mutations, translocations. So basically every type of genetic alteration that we can find in patients, we can probably model using these animal models. The other aspect to consider is the where and when all of these alterations are triggered. So the first version of these models basically were full bodies create or carry these alterations in every single cell of the body. That has evolved a lot, and so we've gone from every cell to making it lineage specific, to making it cell stating specific, and in case of some modern approaches, we can make it at the single cell level specific when and where we trigger these effects. We've come up with reversible versus irreversible systems and constitutive versus inducible. So the pros of using GEMs as a model is that they are fully immunocompetent. So very much like the example of matching a humanized recipient with a primary tumor cells. In this case, we have full myeloid and lymphoid compartments that are fully functional. We have, at least in some model, a fully relevant tumor microenvironment with stromal populations. They are particularly good at letting us assess longitudinal processes and transformation. And we can borrow multiple tools from the immunology field to further enrich these models. Of course, these are not perfect, and some of the consideration we need to keep in mind is that these models tend to be cost, time, and resource demanding. It can take many years to develop a single model. There's, of course, interspecies considerations at the gene marker and cell level between human and mouse. And some of the challenges I'm going to be touching upon, we have to do with the penetrance, the latency, and the detection of the disease. So the use of GEMs in lymphoma modeling is not new. I pulled up this paper that's all the way back from 1985, where this group was already observing what was the effect of engineering overexpression of c-Myc under the control of an immunoglobulin enhancer. But over the years, they have become easier and easier to make, and that's particularly true now with the real explosion of CRISPR as an editing tool. So this is, as of mid last year, some of the multiple alleles that have been engineered to recapitulate genomic alterations in DLBCL. So I'd like to spend the next few slides focusing on one particular model and one particular disease that we, in my group and those of our collaborators, were very interested in. So just to put the general broader context, we're talking about diffuse large B-cell lymphomas, or DLBCL. This is the most frequent form of non-Hodgkin lymphoma. It arises from activated mature B cells, much like NHL in general. The median age of diagnosis is 70 years old. The reason why we care about this is because this is a rapidly expanding age group population, and where very aggressive therapies are poorly tolerated. So while most of DLBCL, so 2/3 of DLBCL patients present disease in their lymph nodes only or in lymphoid tissues, about 1/3 can present tumors elsewhere. And that includes a combination of presenting tumors both in lymphoid tissues and on other sites, but the disease can also occur as a primary event, targeting many different organs and including even immune-privileged sites. So our interest in this form of DLBCL, called extranodal DLBCL, comes from the fact that the 5-year survival rate for these tissues, for this disease, drops significantly as compared to the nodal disease, although this depends a bit on the particular site and stage of the disease. So two large areas of needs regarding this tumor have to do with we can't really anticipate when the spread is going to happen, because we don't really know, or we didn't really know much about what a pre-malignant state looked like. And the other one is, in many cases, the standard forms of frontline DLBCL treatment is not always adequate or very efficient to treat these tumors. They tend to relapse a lot. And we also have to consider the particular context in which these tumors grow, which is very distinct stroma and microenvironment from the counterpart located in the lymph nodes. So what do we know about these tumors or this extranodal disease? So about 5 years ago, investigators, including those here in BC and in BC Cancer, came up with a molecular classification for DLBCLs, depicted here in the different subgroups, based on the combination of mutations that these tumors regularly harbor. So one particular subtype, called the MCD subtype, has one of the poor prognosis among all of the types, and perhaps not surprisingly, it shows a strong link to extranodal disease. The name MCD comes from the fact that these tumors usually harbor activating mutations in the genes MYD88 and CD79B. So we and others set on a quest to say, this is a problem that we care about, this is the information that we have, can we create relevant models to study this disease? So we needed to get a bit more clues on how we could or should construct our models. So from the clinical profiling of these tumors from patients, we had different clues. So we knew that there was definitely a hierarchy in how the mutations are acquired in these tumors, with some being more founder clonal events and some being more secondary events. Based on the mutation burden in these tumors, they appear to have the highest overall level of mutations. Their B-cell receptor, in many cases, seem to be prone to detect autoantigens. So they appear to be a self-reactive nature in these tumors. Based on transcriptional profiling, they appear to be T-cell depleted, so meaning that the cells probably found ways independent of T-cells to divide and grow. And also that there were very recurrent genetic alterations linked to enabling immune evasion in these tumors. So to come up with a model for this disease, we decided to start with what we could control, which were the mutations, and we set our focus on mutations in these two genes, MYD88, one of the defining genes for this, and the other one called TR1XR1, just based on the frequency of these mutations. So when building a model, what could we control? We could control the genetics of the model. So our group around 5 years ago, but also more around the same time, a group in Germany by Christian Reichand and Jared Needle, where we both had the same goal of developing a gem for this disease. So in their case, they were generating a model using or leveraging a model carrying a MYD88 gain of function mutation. We were doing something similar using a TR1XR1 loss of function mutation. So our models had those mutations. They were generated in a way that only B cells would carry these mutations. And to accelerate the process that might otherwise take a really long time, we added BCL2 overexpression in this model, which is anyway a feature that we can see in clinical specimens. So this is the part that we can control of the models. What we couldn't control, but we wanted to see ideally, was that these models led to an aggressive disease that led to the extranodal involvement, that showed a large B cell histology, had elevated rates of mutation, that they could recapitulate the exact mutations that we see in patients, and why not to see what some of these self-reactive features. So to make a very long story short, I'm going to cover some highlights very quickly, but the idea is that both our collaborators and I could see in our models basically these features come up. So if we age this animal, we started to see tumors arising at extranodal sites. We could see that the morphology of large B-cell immunoblastic cells matched that what we would expect to see in patients. We could see that mice carrying this TR1XR1 loss of function mutation or the MYD88 gain of function mutation show much shorter survival. So we're talking about a fairly aggressive disease. By sequencing the B-cell receptor in these tumors, we could demonstrate that there was a very high burden of mutation in these tumors, again, mimicking what we were seeing in the clinical specimens. Looking at mutations overall in the genome of these animals, it was very gratifying to see that beyond the seed mutations that we planted in these animals, if we genotype now the full-blown tumor developing these animals, we're seeing acquisition of additional mutations. And in many cases, these are the same mutation or the same type of genes that we see affected in the human specimens. And finally, if we take or isolated serum from these animals, we could actually see the formation of autoreactive antibodies. So going back to the list I presented a few slides ago, basically every one of the checklists that we could have asked for these models were actually coming true. So additional features that we saw in these tumors, or that are a bit general to GEMS, is that not all animals will develop tumors, or evident tumors at least, even when they carry the mutation. That the disease very rarely happens in young animals, that the disease onset varies in time, the tumors are where they are supposed to be for the most part, and that from one animal to the other, the location of the tumor may change. So all of these are features that we actually find in patients. So this is great in terms of a model that recapitulates the disease, but in terms of research, it presents some additional challenge. So we need to come up with large cohorts. We have to age animals. We have to do a lot of frequent health checking and monitoring for clinical signs, and maybe even include imaging approaches to study where and how the tumors are developed. So the question was, we have these very good models for human MCD. Can we use them to test or guide therapeutics? And that's some of the questions our collaborators started asking. So one example is our collaborators had noticed that there was a collection of patients with DLBCL that show co-upregulation of BCL2 and PD-1. Now, both PD-1 and BCL2 have drugs that allow us to target it, but in phase 1 clinical trials, they have showed somewhat limited responses. So the rationale for this study was, if we see that these effects alone don't have a lot of effects, can we combine them and come up with something better? So what they did was to set basically an animal hospital with doing all of the aspects that I highlighted. So a large cohort of animal aged for more than a year, and they were doing actually frequent MRI scans on all of these animals to test when and where the disease was developing. So once they could confirm the presence of masses, they actually randomized the animals, much like in a clinical trial, and then assigned treatment. And what they could show is that actually while the agents targeting BCL2 and PD-1 by themselves were not super effective, the combination did really make a big difference. So this is an example where using this model could pave the way for now moving this to a clinical implementation or clinical trial at least, and see whether this combination of treatment could work where the single agents fail. These models are also useful for testing immunotherapies. So in these same models, you can engineer autologous T cells. You can isolate autologous T cells and generate something similar to a CAR T cell product. And the group could show in their same model that this can be used, and that the response to the therapies has a lot to do with the initial amount of burden of the disease that the animals was harboring, and that's basically the same that we see in the clinic. So, so far, these models have allowed us to basically test new combinations, and they could be useful for immunotherapy. But the question that was asked was, can we actually guide treatment using these models? So in DLBCL, there was a lot of anticipation and expectation for potentially the broad deployment of ibrutinib, this BTK inhibitor I referenced before. But during the Phoenix trial, which assessed the implementation of this agent in combination with standard of care therapy, they show that at least taking the whole collection of patients, there wasn't a net advantage to include this as a therapy. Now, retrospective analysis of this data, now classifying the tumors based on molecular subtypes, including the MCD category, show that MCD tumors with ibrutinib should be doing very well. So what this group proposed to do is, okay, we know that ibrutinib now may be beneficial to MCD tumors. From their previous studies, they know that combinations with venetoclax and anti-BCL2 agent could be useful. So what they did was, again, do their animal hospital approach, take their combination therapy, and demonstrate that while the single agents were not great or had limited or at least time-dependent effects, their combinations really allow them to achieve long-term remissions. And so they actually use this information to inform the treatment, the off-label treatment of a group of patients that have basically gone through all of their options, so relapsed refractory disease, had failed stem cell transplantation, had failed to see response with CAR T cell. They apply this combination of treatment of ibrutinib and venetoclax, and in 5 out of 6 patients, they saw at least a temporary response in the patient. So the models can actually be used for informing clinical decisions. So I'm going to go very briefly over this last part, just for the sake of time. But another thing that we can get from these tumors is information on where do these tumors originate and how do they come about. So instead of asking how can we treat the tumors, the idea here was to we're asking ourselves, is there or are they B cell populations that we can define as a true precursor for these tumors, and that potentially could have a distinct phenotype that we can track early on? So what we did is to take B cells expressing the MYD88 gain of function allele. These were from healthy young animals, so they were free from disease. And we produced some transcriptomic data to try to understand the difference between wild type and mutant B cells. Now, what we found is that some of the alleles that were upregulated by the presence of this mutation were frequently seen in a very particular B cell type called H or autoimmune B cells. So not only there were particular genes that were found in H and autoimmune B cells upregulated by this mutation, but actually the whole transcriptome was enriched for this. So what are H or autoimmune B cells? So these are cells that are typically found, as the name suggests, in patients with autoimmune disorders. So they're aberrantly expanded B cells. They express the transcription factor TBET, which is most commonly found in T cells. They respond to interferon gamma, TLR signaling, and they're really responsible for the production of pathogenic antibodies. They can also be seen in the context of viral infections and also in infections with, for example, with malaria. But no one had yet made a connection between these and lymphoma. Now, curiously enough, the fact that or the presence of an autoimmune disorder or chronic infection actually increases the risk of developing lymphoma. So this was an interesting lead. So what we ventured to do was to actually demonstrate that in addition to a transcriptional profile, these mutations, these founder mutations for NTD could actually lead to the expansion of the cells, and we could effectively demonstrate that they were expanded in young animals. We could also demonstrate that a different founder mutation in these same tumors led to similar effects. So we have an overexpansion of these cells that should not be there in the first place. And then we venture, this led us to the idea that these cells, or cells carrying an H or autoimmune-like phenotype, could be potentially these precursors for MCD lymphomas. So what we showed, and to make a very long story short, is that over time, we could see a disproportionate accumulation in the animals that eventually moved on to develop tumors. We could show that the clonality of these B cells was the highest among other B cell populations in these animals. And moving to human clinical specimens, we could show that at the transcriptional levels, patients with advanced level of disease that usually correlates with disease dissemination had the highest level of this master transcription factor in these cells, not only at the RNA level, but also by IHC when looking at FFP samples from patients with this type of tumors. We also extended this data and showed that leveraging publicly available data from an independent study that cells with this particular phenotype exist in human tumors. So as a partial summary, and I'm almost close to my last slide, these gems that we and others developed actually recapitulate key anatomical, histological, and biological aspects of human DLBCL. They can be used both for testing conventional treatment, but also immunotherapies, and they can also provide a lot of information of the origin of the disease, paving the way for early stage detection. So on a more general note, and now, yes, I'm coming to the end of my talk, that's a take-home message for everyone today. From the first section is that inter-intratumor heterogeneity are largely responsible for most of the challenges that we still have in the clinical management of lymphomas. That preclinical modeling can complement and inform, but definitely do not replace clinical studies, but they can work side to side. And GEMs in particular are a great platform to study disease pathogenesis and to advance immunotherapies. So finishing off with these additional phrases, and it's used to move on to my last slide that has to do with where are we moving forward, or where is the field currently at. So some of the aspects that we and others are working very actively on have to do with, well, on the one hand, generating better and more and to implement tools for gene editing, models for viral system delivery to facilitate the generation of these models. There are a lot of groups working on generating even better version of the humanized mouse models. There's a lot of investment in using, for example, microfluidic devices to come up with better organized systems. And in the GEMS field, which is something that my group works on, we're having a lot of effort invested in coming up with models that can better recapitulate the timing of the mutation when you have multi-hit models. So if we have 2 mutations, they're probably not going to happen at the same time. So that's something that we are trying to consider recapitulating our models. Other thing that we're putting a lot of effort is beyond the mutations, trying to generate an immune context or history that's relevant to the disease or for each type of disease. And finally, to develop new genetic engineering mouse models for rare forms of lymphoma. So with that, I know I'm on the hour. I'd just like to pay my acknowledgements to my lab, collaborators, and the CLC and facilities of DC Cancer funders. And if there's time, I'm happy to take any questions.
Any questions for Leandro?
I know it was a lot, so.
Well, if there's no questions, thank you, Leandro, for your wonderful presentation and beautiful work. That's excellent, excellent job. Thank you.
Thank you very much again for the invitation.
Yeah, thanks everybody. Have a good weekend.